Now, so just going back to the pathology we were talking about before, we have these things in the body called tumour suppressor genes. And that's what they do. They're going to inhibit cell division and inhibit the formation of tumours and cancers. So having healthy, active tumour suppressor genes is going to greatly reduce my chances of getting cancer. So I want my tumour suppressor genes to be as active as possible. And the idea is that if the spike protein can inhibit the activity of the tumour suppressor genes, then what is left to suppress the tumour? And you mentioned the specific examples there, MHS3, P53, and I think BRCA2 is probably another one, that BRCA, breast cancer.
I saw it mentioned in a review. I hadn't seen the primary data on this, but that's a suppressor gene. That's the one associated with breast and ovarian. So people who have BRCA mutations are much more likely to get breast and ovarian cancer before they're 50 than those that haven't, which really upholds the model of what I was saying. Interfere with these things and your cancer pops out a lot earlier. And that's what I'm worried about.
So these suppressor genes are exactly what we want to be healthy, but they are potentially being inhibited. And the other thing the paper pointed out was that the modified mRNA vaccine contains N1 methyl pseudouridine. Now, this is one of the bases in RNA, and it's not the natural uridine, it's pseudouridine, because it lasts for much longer. And that means the spike protein could be produced for a much longer period of time. Again, potentially going on to inhibit more activity of the tumour suppressor genes. It really is quite a frightening potential scenario.
It certainly is. Now, again, when I pointed all this out, people say, "Shut up! You're just a cancer doctor. You don't know what you're talking about," and that really gets me wild, as you know from all my HIV experience. But one of my other things, I've been on many scientific advisory boards for grants, authorities, and companies. And I was on the scientific advisory board for a company that, after its name, it says the messenger RNA vaccine company. I was on that board for 5 years, and I knew there was a big problem because messenger RNA gets dissolved and killed and mopped up in 20 seconds if it's in the body. So you can't possibly use it for a vaccine until you stabilise it. And the very fact, if you stabilise it, the big problem is it's stabilised. So that is the fundamental flaw with RNA vaccines.
And carries on for an indeterminate period of time.
Banned. I mean, no boat. They should be completely banned. If they're going to be used for anything, it has to be what they are, gene therapies. And you might use those in advanced cancer and things like that, but you should never use them in general population as vaccines because they're not.
Yeah, because in advanced cancer, someone's life is already at risk, so we can carry out more risky treatments, potentially. It's all about risk-benefit analysis. The paper also talks about suppression of cancer immunosurveillance. What is cancer immunosurveillance and how could it be suppressed?
Well, I think that's what I was mentioning about the T cells. Yeah. It's just suppressing the T cells, and that's what they do. And if you do things that suppress the T cells, up you pop with cancers. And the ones you're most likely have got along that progression line, sitting at 5 out of 6, they're the first ones to pop up. And HIV showed us that. It was just an incredible model. So you suppress the T cell surveillance, up pop the lymphomas, because the lymphomas associated with EBV, Epstein-Barr virus. Everybody is infected with Epstein-Barr virus. Only about 1%, far, far less than 1% ever get a cancer with it. But what that did is remove the T cell suppression, which is T cell surveillance, which was keeping it in check. And the other one that popped up was Kaposi's sarcoma. I mean, that really popped up massively in HIV, and there wasn't a virus associated with it. But they thought there must be, and they found it. And that's called human herpes virus 8. And left to its own devices, it does nothing. It will induce change, some nasty changes in the skin, etc., but it won't kill you. But you lose your T cell ability to suppress that virus, and it comes out with great angiogenic capillary sarcoma that has actually throttled people. I mean, it's so out of control, it'll actually throttle them so they can't breathe. I mean, it's just unbelievably horrible. And that's never been seen until the HIV came along. But we know the principle because you see these diseases and skin cancer, what have you, in patients who are heavily suppressed and long-term suppressed for transplants, etc. And then you have to modify that, etc. So that's what is meant by the immune surveillance. It's purely the T cell and the innate immune system that are doing it. And what it tells you is what I've been arguing from the beginning of my oncology career is the most important thing in controlling cancer is the immune system. And I was always told, no, it's not. No, it's not. It's you have to kill them with toxins. I mean, everything was chemotherapy or radiotherapy, and the result was all the same as, total destruction. It didn't entertain any role for the immune system whatsoever.
I mean, I would imagine at our kind of age, I probably get cancer most days, do I? Is that reasonable? It's just the immune system keeps it down or eradicates it?
Most people speculate, people go into this, and some mathematicians sort of even reduce it to the number of cells that might mutate arise in your body every day. And the T cell system controls them. And I mentioned before that I think that it is the innate immune system that controls them, as opposed to the highly adapted one, which vaccines are meant to induce. And that the thing that concerns me, and if I was Chief Medical Officer, I'd want to do something about it, is that highly important innate immune response falls off when you're about 55. It really starts giving up. And by the time you're 70, 75, it's in your boots. And if you transform a figure for the cancer instance of that age range, it is on the floor about 50, 55, and it goes up and up and up and up to your 70, 80. It's not necessarily causal, but the association is so tight.
And it's a perfect inverse correlation, isn't it, as the cancers go up?
So
in exactly the same way the T cells go down.
In fact, when you draw it, it looks like a butterfly wings.
Yeah.
So obviously, to help control cancer, the best thing to do is try and boost that innate immune system as it declines. It's involved in everything, with your susceptibility to infections, bizarrely cardiac disease, but definitely cancer. And doing something about it and addressing that, I think, is one of the most important things that a chief medical officer could do, but they don't seem to be interested. We do know, and you've had other guests here who've told you the most important thing everybody can do, and the government should do it, as everything they orchestrate and conduct, they seem to make a mess of. Vitamin D supplementation to good high levels is the most important thing that can be done to stop the innate immune system doing. They don't do anything about that. And when they do come out with some inane comment, they say, well, all you need is 400 international units for
nothing.
Your bones. No, we're not talking about bones. We're talking about the immune system and the surveillance of these other diseases. That does really annoy me. Then the other things, it can be stimulated with mycobacterial things of various descriptions, and I focused on a heat-killed mycobacterium, which I've discussed here before. Now that is so easy to make and do that I would again look at that. They put so many hurdles, and I noticed there was something in the paper only this weekend that it's still impossible to get for small companies to get sensible things through, for cancer. The hurdles are so high and so prolonged, costs a fortune. This is why these companies go and collapse. Yet you come along with something like an unproven, untested, with a heritage that every time it's been tried in any other form, it's been a disaster, and all the things they get, we want to make sure it's safe. And we must have more. This is what they told me. They told me about that. It needs more work in animal models.
Yeah.
This is after it's been in patients, hundreds of them without a single side effect. Yet they didn't seem remotely interested about the side effects or safety of the vaccines that they poured out. It's completely discombobulated. I personally think the MHRA is unfit for purpose and should be completely shut down, as I do with the Department of Health and the NHS that run it or meant to regulate that too, because it's been an absolute disaster. It's a disaster for what we've had to go through with the vaccines, but it's a disaster is that they seem to make it so difficult for anything that is effective, safe and cheap.
Yeah,
they're not interested in. You've got
you've used this. It's basically a BCG type preparation, isn't it, with bacteria, and you've kept patients alive with this for decades. It boosts T cells, and yet, if it was available, I would get the train or drive down there to you now and be injected with it, because I'd love to boost my T cells. But the Medicines and Healthcare Products Regulatory Agency won't license it for compassionate use. It just seems outrageous.
I think
I'm being deprived of. You could boost my T cells tomorrow, and yet you're not allowed to. That's just outrageous.
They won't let the company release it for compassionate use or off-label use, when that has been a pattern which used to exist, but it's under the guise of safety, when there are literally thousands of people being given this and similar things in TB trials with no side effects whatsoever, yet they bang on about side effects and safety issues and stuff. It is a farce. It is a total farce.
And if you were allowed to boost my T cells, I'm less likely to get a whole range of cancers, probably get less infections.
Yes.
And it's safe. And in fact, to call this a vaccine is misleading, because this is giving some borate buffered, from what I remember, heat attenuated bacteria. And to use the same title for that as these wretched mRNA genetic concoctions, to me is quite an outrageous conflation.
We actually call these the category they fit in as immune modulators.
Yes.
As opposed to vaccines, because vaccines now got a really dirty name.
Yeah.
And the COVID vaccines are COVID. What BioNTech says on their first sheets, they go COVID vaccine and gene therapy. They actually have gene therapy written on there. I was staggered. It's that blatant in your face.
Yeah. Yeah. I've just got really one more question in mind. From your experience, what blood levels of vitamin D are desirable in cancer management and cancer prevention?
Over 100 nanomoles per litre, which is twice the level that the NHS say is normal.
So that's about 40 nanograms per mil, isn't it? Roughly that in American. Over 100. That is really useful, actually. So GPs around the country should be testing this and titrating everyone up to that level, I would have thought.
I first picked this up when I was doing very early cancer vaccines, and then we had the immune modulators. Vaccine implies it induces a particular immune response against a certain particular antigen. That's a vaccine. Now, the IMM mycobacteria, they don't do that. They induce an activity of the innate immune system that then allows a T cell to see something that it hitherto hadn't seen. It's like putting glasses on it. And so you then see it, and you get a knock-on effect, like domino effect, by doing this. The problem with cancer vaccines, as I pointed out, and I was a very early, a big fan of it, and I very quick to want to know what works and what doesn't work, is cancer vaccines only work for a few months. The simplicity is that when you get resistant to chemotherapy, the tumour gets resistant and goes on another path. The same thing happens if you immunise it against a certain antigen. Of course, we can control them in mouse models, and you'll see improvements in the humans, but they only last 3 months or so, 6 months at the most. So what's the point of that? You want something better. You might be able to use it for a focused, targeted beating up of a residual cell, but if you don't do it within a certain time, the tumour is just going to mutate and escape. So that is why we've got to be very careful with the word vaccine, because in both cases it's very misleading.
Yeah, but if we use one of your immune modulators, that's not specific. That's going to boost the body's own T cell responses, and the T cells are already pretty smart. There's just not enough of them. You know, the T cells are part of creation. They're brilliant. So, you know, whether you believe they're created or evolved doesn't matter. They're amazing physiological cells that can do all sorts of things. We need to boost them a bit.
Yeah, without them you die. Yeah. And as you get older, they give up before you do. So it's a logical thing to give them a bit of help. Yeah.
Well, as soon as this immune modulator to boost my T cells is available, I'll be down there like a shot. And I would appreciate you giving me one.
We need people to lobby the MHRA and whoever is meant to control them.
We really do.
We really do, because they're not doing their job on anything at all.
It's outrageous. And it's also very consistent with Professor Clancy. I know you're familiar with his work in Australia, giving immune attenuated bacteria that then goes to the pairs factors and goes off to promote mucosal immunity.
Yeah. It's the same principle, just different ways of doing it.
Yeah. So I think practically... people need to, we don't want to frighten people, but we do want people to get treatment as early as possible if cancers do develop. So I think really it's a good idea for people to be aware of potential cancer signs in themselves and their loved ones, and really go to the doctor as early as possible if you have anything that you are uncomfortable with, and get early treatment, because that's going to massively influence the outcome of the disease.
Indeed.
Yeah. Professor, as always, fascinating, mind-blowing, a little perturbing, but the thing I've really enjoyed and benefited from on this talk was the complete integration and harmony between your medical experience and the science. They just mesh together beautifully, which of course to me means that both are much more likely to be correct, because they have complete consistency. So it's a Sunday afternoon, you've given me a lot of your time, and we're very grateful, and thank you very much.
Okay, thank you. Bye.
Thank you.